The light-dependent reactions begin when light is absorbed by chlorophyll, a pigment in the thylakoid membranes of the chloroplast. This light energy excites the electrons, causing their energy level to increase. The high-energy electrons move down the electron transport chain where they attract a proton from the stroma of the chloroplast to the inner thylakoid of the chloroplast. These high-energy electrons are then re-energized soon afterwards. The electrons are then picked up by the electron carrier NADP, which combines within the electrons and a proton to become NADPH. So why doesn't the chlorophyll run out of electrons? Well, it's because enzymes on the thylakoid membrane break up water molecules into 2 protons, 2 electrons, and oxygen. The protons and the electrons are used in the light-dependent reactions, and the oxygen exits the cell. Coming back to the protons, the only way for them to get out of the inner thylakoid is through a protein called ATP synthase. Now, as protons pass through ATP synthase, it rotates, causing a phosphate group to bind with ADP, a low-level energy, to create ATP. The light-independent reaction, or the Calvin cycle, begins with 6 CO2 molecules from the atmosphere entering the cycle. These 6 CO2 molecules combine with 6 5-carbon molecules in order to create 12 3-carbon molecules. These 12 3-carbon molecules are then converted into higher energy forms, with the energy coming from high-energy electrons from NADPH and from ATP. 2 of the 12 3-carbon molecules are set aside from the rest and used to form various 6-carbon molecules, but the remaining 10 3-carbon molecules are converted into 6 5-carbon molecules, which are then combined with 6 molecules of CO2 to begin the next Calvin cycle. In glycolysis, 1 molecule of glucose is broken down into 2 molecules of pyruvic acid. Now, in order for this reaction to occur, you have to put in 2 ATPs, but you get 4 ATPs out of it. So essentially, you're gaining 2 ATPs from glycolysis. One of the reactions of glycolysis involves removing 4 high-energy electrons and placing them in NAD, an electron carrier. Once a pair of electrons is placed in NAD, it becomes NADH. The purpose of NADH is to keep the electrons until they can be passed to another molecule. While oxygen is present, the step right after glycolysis is the citric acid cycle, or the Krebs cycle. But if oxygen is not present, then after glycolysis is fermentation, which we'll get to later. The Krebs cycle begins with the creation of citric acid. Basically, pyruvic acid from glycolysis comes into the Krebs cycle. 1 atom of carbon from pyruvic acid gets added to a molecule that creates CO2 and is released into the atmosphere. The other 2 carbon atoms are combined with coenzyme A to create acetyl coenzyme A. Then the 2 carbon acetyl group is added to a 4-carbon molecule, creating a 6-carbon molecule called citric acid. As the Krebs cycle progresses, citric acid is broken down and 2 carbon atoms are used to create CO2 and are released into the atmosphere. The remaining 4-carbon molecule is used to combine with the next 2-carbon acetyl group to create citric acid again. In one iteration of the Krebs cycle, 5 pairs of high-energy electrons are caught by 5 electron carriers, 4 NADH molecules, and 1 FADH2 molecule. Electron transport begins with high-energy electrons being dropped off at the electron transport chain from NADH and FADH2. These molecules then change back into their uncharged forms, NAD and FAD. These electrons are then reunited with protons and oxygen in order to create water. Now, as these electrons are moving down the transport chain, they attract protons, and similar to the light-dependent reaction, these protons pass through ATP synthase, which binds a phosphate group with ADP in order to create ATP. When oxygen is not present in the cell, fermentation follows after glycolysis instead of the Krebs cycle and the electron transport. Fermentation is different because it allows energy to come out without using oxygen. However, you can't get as much energy from fermentation. Fermentation involves passing high-energy electrons back into pyruvic acid in order to turn NADH back into NAD. This allows glycolysis to continue producing a steady stream of ATP. Since this process does not require oxygen, it's said to be anaerobic. There are two types of fermentation: alcohol fermentation, which forms alcohol and CO2 from fermentation, and lactic acid fermentation, which forms lactic acid from fermentation.